I would like to thank the organizers for inviting me to speak at this Human Technology Conference. And the topic of my presentation will be the gut-immune connection. It's a topic that I recently covered extensively in my new book with the same title. So let me start out with the conceptual model that I have been using for some time now to explain the interactions, the complex interactions between the brain, the gut microbiome, and the immune system on the one side, and the environment on the other side. It is a systems biological view, taking into account the bidirectional interactions between all the involved nodes in this network. On the one side, the gut with its microbiome, the brain, the immune system being a mediator between the gut microbiome and the brain, but also this very important connection to the environment, both in terms of the psychosocial support that the environment provides and stabilizes these interactions within the microbiome system, but also in terms of being a source for both pathogens and antibiotics, so stressors on the system. And I like to continue this concept of the this systems view, that we can understand many of the problems that arise health-wise come from two major perturbation of the balance within the system. One are psychosocial stressors that act primarily on the brain, and dietary stressors that act primarily on the gut. So there's been a dramatic change in the view of the gut microbiome during the last decade. On the left side, you see something that we have known for some time, namely of these many metabolic functions, capabilities of gut microbes, for example, breaking down complex fiber molecules, including resistant starch, into short-chain fatty acids, which are sort of a major currency of microbial communication. But there's many other things that microbes can metabolize, break down from unabsorbable large molecules that we get with our diet into smaller molecules which then can be absorbed, but also can have an effect on our body. If you go to the right side, this is the more recent insights that we've gained, that many of these metabolites and components of the gut microbiome can actually affect pretty much all the organs in our body and organ systems, including the nervous system of the gut, the cardiovascular system, the brain, the immune system, topic of this presentation, but also when I come to the end of my presentation, has some effect on the pulmonary system. So we've gone from a conceptualization of the gut microbial importance from the generation of metabolites in the gut to a system-wide communication system throughout our bodies. And let me just give a quick view of the kinds of communications that have been elucidated. I will not have the time to get into any of the details. We know that there is a communication between gut microbes and some of their metabolites, like again, the short chain fatty acids or metabolites of amino acids that we ingest with our diet, modulating the immune system and, for example, playing a role in the susceptibility and also the resilience to inflammatory conditions of the gut, such as inflammatory bowel disease. Another important implication of the interactions of gut microbial metabolites and cell wall components is with the immune system of the gut in terms of playing a role in the recent rise of food allergies and food sensitivities. So for example, the short-chain fatty acids have a inhibitory effect on these inappropriate immune reactions to certain food components. There's also important signaling to the liver, which I'll not get into right now, but disease-relevant alterations in this communication have been described. Very important cardiovascular disease, certain metabolites of the gut microbes, like this a substance called TMAO, has been identified as a major risk factor for cardiovascular disease, but also inflammatory influences, immune influences on the coronary vasculature, playing an important role in development of coronary disease. And then finally, the important connections with the brain. Again, the main players being the one side immune mediators, on the other side short-chain fatty acids, which have as in general, an anti-inflammatory effect. So it's just a snapshot of the complex interactions that have been unraveled between the gut microbiome, the immune system, and many or most of our organ systems. The gut microbes are quite an intelligent part of planetary life. They have up to 20 million microbial genes, outnumbering by far our 20,000 human genes. And there have been interesting speculations that, have we been neglecting 99% of all this genetic information in a system where we live in close symbiosis with the gut microbiome. So the term holobiome has been coined, meaning that we're really one organism, and this organism is regulated by genes. The human genes, just being like this red line, is represented by this red line on the surface of this human figure, whereas the microbial gene is much more expansive and has a lot more function, many of which we don't really know. So what are the signals that microbes can generate with this vast genetic storage of information that they have? Well, there's basically three types of these signals. Some are diet-derived, some are host-derived from molecules that our body produces, and some come from the microbial, from the microbes themselves, the microbial membrane, which has molecules in it that interact with our immune system. And let me say a few things about both the diet-derived and these microbial membrane-derived molecules. So in terms of the diet, this is illustrated here on we consider a healthy microbiome-friendly diet, which is predominantly plant-based. These nutrients, tens of thousands of molecules and phytochemicals, are metabolized by this vast number of microbes we have in our gut. You can see this generating a vast number of metabolites and signaling molecules, which then interact with our body and the different systems in our body, including the immune system and the brain. And let me just illustrate one of these examples. Just step back here again. One of these examples of a diet-derived signaling system that the microbes play a key role in. So this has to do with the essential amino acid tryptophan that we have to eat with our diet because our bodies cannot produce that molecule. And we know today, in just the last few years, that tryptophan can be broken down into serotonin, represented here with this abbreviation 5-HT, 5-hydroxytryptamine, and it's stored in cells in the gut. 90% of all the serotonin in our body is stored in these cells in the gut, and these cells communicate closely with the brain through the vagus nerve. But what's interesting is that this synthesis of serotonin is to a great degree influenced by signals from the microbes, again, the short-chain fatty acids or secondary bile acids that stimulate a hormone in these cells to metabolize tryptophan into serotonin. There's another, so if serotonin is the good guy in this tryptophan metabolism, this microbial enhanced or mediated tryptophan metabolism, another group of molecules, the kynurenines, are the bad guys. So those kynurenine molecules can get into our central nervous system and into the brain and cause neuroinflammation and neurodegeneration. Again, we know that microbes, for example, it's been shown in animal models for lactobacilli, play an important role in this conversion of tryptophan to kynurenine. And dependent on the composition of the microbes in the gut, we get more production of serotonin or more production of kynurenine. So it's a yin and yang that comes out of this tryptophan metabolism. And then there's a third group of molecules that microbes play the key role in metabolizing, because our body does not have the molecules, the enzymes to break that for this particular pathway. And these are the indoles. The indoles, it's a group of molecules that can have both beneficial and detrimental effects on the brain. One particular member of this family, indoxyl sulfate, has been identified as an important player in several in autism spectrum disease and in Alzheimer's disease. So this is just illustrating the complexity with one molecule, tryptophan, our diet, that the microbes play crucial roles in generating signal molecules that affect the rest of our body. Now, let me say a few specific things about the gut microbiome immune interaction. Again, because of lack of time, I do not have time to get into the details of well-studied systems, how the microbiota interact with both receptors on, for example, on immune cells that have extensions into the gut lumen and can interact directly with microbial cell wall components. But ultimately, this interaction between the microbes and the gut-associated immune system has major effects on the immunity, not just in the gut, so defenses against infections, inflammation, but also throughout our body. And this is illustrated here, figure from an excellent recent review that shows that immune signaling plays an important role in establishing this gut-liver connection. So immune modulation in the liver originating from these interactions of microbes in the gut with the gut-associated immune system. Then the gut-brain axis, the interactions and the signaling to the brain of these immune mediators. And also, and we'll come back to this at the end, a concept that has only recently sort of been emphasized, this gut-lung axis. And I'll come back to these various connections that come from interactions of the microbes with the gut-associated immune system, which then establishes communication links to all our other organs and plays an important role in the health of these organs. Let me just illustrate this in one figure for the brain. So these cell wall components of certain microbes with this abbreviation, these molecular patterns that are cell wall components, such as lipopolysaccharide or flagellin or others, which activate receptors, so-called toll-like receptors on certain immune cells in the gut, particularly initially the dendritic cells, which then start producing cytokines. These cytokines can spread throughout the body, affect all the other organs, as I just said, but they also can cross the blood-brain barrier, activate immune cells in the brain, the microglial cells, which then amplify this inflammatory cascade. And these immune modulators, particularly cytokines, can then affect the activity, structure, and function of nerve cells, leading to neuroinflammation and neurodegeneration. So something that 10, 15 years ago, we didn't even conceptualize at all. There's something that originates in the gut, particularly with beneficial microbes in our gut, that if they get too close to immune cells, that it triggers a cascade that ultimately could play a role in some devastating diseases such as Parkinson's disease and Alzheimer's disease. And we know a lot of details from animal models, which cells in the brain, so from nerve cells to these various glial cells or endothelial cells that make up the blood-brain barrier, how they can be affected in a negative way by these cell wall components of microbes in the gut, but also in a positive way by these short-chain fatty acids. So these small molecules that are generated when microbes break down complex carbohydrates that they get in terms of dietary fiber in our diet. So a concept that has received a lot of attention in the lay media, but also is now kind of accepted in scientific jargon, the leaky gut concept. And this is a very important concept, explaining some of these inappropriate interactions of our gut microbiome, which is a healthy symbiotic ecosystem with our immune system and the negative consequences. So on a healthy diet, we have a large variety and diversity of the gut microbes. They are separated from the gut-associated immune system by two mechanisms. One, a mucus layer that does not allow these microbes to get close to these dendritic cells with their sensors, and then with a very tight epithelial barrier where these cells that make up the gut lining will not allow for any components of the gut microbes to get through and come in contact with the gut-associated immune system. So what happens in the leaky gut situation? First of all, a leaky gut can be the consequence, or is the consequence, of a chronic dietary and psychological stress situation. Both of these stressors or perturbations of the homeostasis can affect the system in a similar way. There's a decrease in the abundance, richness, and diversity of the microbes in our gut under these conditions. There's a narrowing of the mucus layer that separates the microbes from these dendritic cell sensors. And once that happens, there's an immune activation, cytokines are being released, and the tightness of this epithelial layer decreases, and now entire microbes can get through into the systemic circulation, so this thing called translocation, resulting in a condition of inflammation, a low-grade systemic inflammation throughout our body, something that has been called, if it originates from an unhealthy diet, it's called metabolic endotoxemia. And there's no infection, which you normally need for getting an endotoxemia. It's really just related to these dietary or psychological stressors. And that metabolic endotoxemia, in combination with a genetic risk that somebody may have, can play a big role in the increase in the likelihood of developing any of these diseases that at first glance you wouldn't think are really in any way related to each other. Give you one example, what we think has happened in the last 75 years or so, after World War II, with the accelerating industrialization, westernization in the world, changes in lifestyle, the early colonization of our gut with microbes related to antibiotic exposure, a lot of exaggerated hygiene measures, and then medical practices, particularly again, antibiotics. All these have led to a change in this gut microbial ecosystem. And as I said earlier, in general terms, the changes are in decreased richness, abundance, and diversity. So the ecosystem has suffered in its resilience and resistance. But this altered gut microbiome, which has adapted pretty rapidly to these changes in 75 years, now interacts with our ancient gut immune system that is largely genetically determined, and that will generate responses that are really optimized for acute immune responses, not for this chronic mismatch between an altered gut microbiota, altered gut microbiome, and our gut-based immune system. So the consequence of this mismatch between these two is that now we have, as I've said several times, a spread of the immune activation throughout the body, affecting a variety of organs throughout. And so I like to sort of recapitulate this, because this is a key point in my presentation and the way I look at the brain gut microbiome system. So we have this bidirectional interaction between the gut microbiome, the brain systems, and the gut-associated immune system. This system is exposed to two major perturbations: brain-targeted stress from the psychosocial stress we are exposed to in terms of a chronic threat to the homeostasis of that system, and also the same with a chronic dietary stress in terms of our unhealthy diet. The end result of this is that it changes the gut-associated immune system with consequences throughout the body. So what has happened, again, over the last 75 years, and something I elaborate on in my books. So these chronic stressors, dietary and psychological stress, can be referred to as an increased allostatic load. Allostatic load means when a chronic stress constantly challenges these stress response systems, the neural system and the immune system. And instead of providing an adaptive response that protects us from these stressors, the stress response itself will become maladaptive, as shown in this chronic engagement of the immune system in an inappropriate way. And so here you can see these effects of the, called the allostatic load, gut microbiome alterations, chronic systemic immune activations, metabolic alterations, negative effects on many organ systems, including structural and functional changes in the brain, and this epidemic of chronic non-infectious diseases that we're experiencing. And I count all these diseases in as part of this chronic non-infectious disease epidemic that we have been seeing and see increasingly now in the developing world with a westernization of lifestyle. All these different, seemingly different and unrelated diseases that are all common, have increased in prevalence, and now affect younger and younger patient populations, can all be related to this inappropriate immune activation. And this has led to a situation that is quite worrisome. So if you look at the distribution of the population from individuals that are in a state of optimal health, less than 5%. If you look at the percent of population that is in a recognized chronic disease state, 45%. But then we have something in between, which is this pre-disease state or suboptimal health state, where this increasing allostatic load with its increased systemic immune activation is already present. No disease has been diagnosed. Patients may be asymptomatic, but they are on their way to this chronic disease epidemic. And so this, we can say, puts gut health and the gut and its interactions with the diet and its the immune system responses, puts this at center stage for this chronic disease epidemic. Again, shown here in red, the links that we can attribute to abnormal immune system activation. And I've added one more to this, which is not a non-infectious disease, but it's this vulnerability to viral disease that has become apparent during the pandemic and during the research that has been done on vulnerability to COVID-19 and particularly abnormal and more severe disease courses after infection. So there's been a very interesting paper from the Chinese University in Hong Kong, and the summary of that paper, so the title was Gut Microbiota Composition Reflects Disease Severity and Dysfunctional Immune Responses in Patients with COVID-19. And just as a reminder, COVID-19 affects primarily the lungs. It's not a disease that goes through the gastrointestinal tract primarily. So this is something where the gut affects a distant organ. And the summary of this paper showed that gut microbial composition in patients correlates with disease severity and plasma levels of systemic immune markers. The same markers that I said earlier are part of the systemic immune activation in all these, in this chronic disease epidemic. COVID-19 patients had lower abundances of gut microbes with known immunomodulatory potentials, such as F. Prosnitzi, Eubacterium rectali, several bifidus species. What they have in common is that they are producers of short-chain fatty acids, of particularly butyrate, that have these anti-inflammatory effects. So a decreased number of bacteria that produce these protective molecules. And in this study, they showed that this altered gut microbial composition in COVID-19 patients persisted after clearance of the virus. There's a lot of things that could be discussed about this paper, but in view of the limited time, I will have to defer this. But to illustrate it in a visual form, so the effects, the changes in the gut microbiome that most likely were present in these individuals before they got infected in response to an unhealthy diet and chronic stress. Again, we have quite a bit of evidence that the patients particularly vulnerable to these more severe infections are under greater psychological and dietary stress, and that these changes in the gut then can affect the immune response in the lungs, and that may also play a role in some of these unexplained brain alteration or brain pathologies that have been described in some patients with COVID-19. Also, this entity of long COVID, this persistence of fatigue, brain fog, and cognitive dysfunction that so many people that had the infection, even if the infection was mild, complain about. I would like to speculate that they also are related to the effect of the exaggerated immune system activation in response from the gut microbial immune interaction, play a role in this as well. So there's lots of research that will have to be done still to corroborate some of these concepts. But I think it's pretty clear that the gut microbiome and its interaction with the gut immune system play a crucial role, not just in this chronic non-infectious disease epidemic, but also in the vulnerability to complications from COVID-19. So I would like to end this presentation with emphasizing the importance of human nutrition. We don't have medications at this stage or interventions that are able to target specific changes in these altered interactions in the gut. But we do know several important aspects of the story which have consequences of what we eat and how we look at our diet and our nutrition. So in this excellent review paper by one of the pioneers in the field of microbiome science, the key statements that they made, that dramatic changes in socioeconomic status, cultural traditions, population growth, and agriculture in the last 75 years are affecting diets worldwide. Understanding how our diet and nutrition status influence the composition and dynamic operations of our gut microbial communities and the arms of our immune system represents an area of scientific need, opportunity, and challenge. It's my prediction that this will be a major focus of research in the next 20, 30 years. And these insights and the solutions that come from this will help to address and hopefully come up with solutions for a number of urgent global health problems. And let me end with a few take-home points from my talk. First, communication between the gut microbiome and the gut-based immune system is an ancient, evolutionary conserved system that modulates the function of every organ, including brain structure and function. So the system itself is ancient, goes back in evolution to hundreds of millions, probably billions of years. What has gone wrong is what happened in our environment and how this system has reacted to this. This ancient communication system connects the mind, the gut, connectome, food, and environment in a way that can not only best, but in my opinion, only be understood in a systems biological way. And key components of plant-based diets act through the gut microbiome to reduce systemic immune activation and the prevalence of chronic infectious diseases. I would add to this, they may also, these components such as the short-chain fatty acids may also play an important role in protecting us against both the vulnerability and the most severe consequences of the viral pandemic and COVID-19 infections. So as I mentioned, I have recently published a book, The Gut-Immune Connection, which deals and gets much deeper depth of the information I presented to you in this short talk. You can either order the book or you can go to my website, sign up for our Mind-Gut Connection newsletter, or get a lot of information that we provide on this website on these various topics. So thank you very much again for allowing me to give a presentation at your conference. And I hope this will have been beneficial and interesting to some of you. Thank you.